our next speaker needs no introduction. So Jay Kizhakkedathu is a long-standing member of the CBR. He's a full professor in the Department of Pathology and Laboratory Medicine, an associate member of the Department of Chemistry, and an associate member of the School of Biomedical Engineering. In addition to all of his numerous accomplishments, he's a wonderful colleague, and we're very fortunate that he is here to speak to us about next-generation blood storage devices. Jay, thanks.
Good morning. Can you hear me? Okay, good. Okay. Thank you for the introduction and also the invitation. Today, what I'm going to do today is kind of like talk about one of the projects in the lab in transfusion area. So we are looking at to develop kind of like better or improved blood bags, see whether actually we could improve the blood storage or some of the problems or some of the challenges faced by the transfusion people actually can be addressed that. So here, so disclosures for this. So I'm not talking any of those in any of these companies. This work is not related to any of these companies. So this, the coding in the project in the lab actually started by Kai and Jan, and then currently actually is done by the couple of graduate students, especially Nicholas's work actually I'm going to talk, and then Parisa and Jose is also working on this project. And Hussein also kind of involved early, and then these people graduated, and Kai is still with us, and these people are still graduate students working in the lab. So why we are looking at actually, like we already heard about kind of transfusion and what is the importance of that. Actually, you can see that this is an example of a platelet transfusion. I can see that kind of like a lot of kind of different emergencies actually deal with the platelet transfusion. And also it's chemotherapy, ICUs, and surgery actually like platelets are used. And also similarly actually like red blood cells or whole blood, these are potentially used there. And there is a lot of kind of like use for this, but and what are the challenges? So if you look at the challenges, actually there are many different things are not addressed or currently actually like the blood services are facing these challenges. So one of the things actually talked about kind of like storage conditions. It could reduce the platelet shelf life and also kind of the RBC shelf life. So if we can improve that, actually that is going to be kind of like really good. Usually like platelets are stored like 5 to 7 days. In Canada it is 7 days, and it's because of kind of like several reasons. Not only the shelf life, the platelet quality deterioration, but also some other reasons. Another problem is kind of the bacterial growth or the, contamination. Actually, like contamination could problematic actually. So that is the reason. For example, in platelets stored at kind of like 22 degrees at room temperature, so you have a kind of a better chance of bacterial growth actually. So because of that, actually, like the transfusion time is limited to either 5 days or 7 days actually. So you have to transfuse them before that. And also that's I mentioned about actually, and because of all of these reasons, actually, like there is a wastage of blood products. Actually, these are precious donated products. Actually, usually 20 to 30% of the, for example, platelet products are kind of wasted, and this and we thought actually like this is not the bacterial contamination, but the storage life or the storage deterioration is something to do with the bags. Currently, actually, like these bags are made of kind of polyvinyl chloride, and it's a hydrophobic, and so we thought actually like storage or the platelets interacting with the blood cells interacting with the bags is kind of like deteriorating the blood cells quality, and that is kind of like one of the reasons possibly we can address. And in terms of like bacterial contamination, there are some technologies available currently. Actually, for example, in platelet storage, pathogen reduction technologies, actually, which are UV-based technologies. Actually, 3 technologies are available currently, and they add certain organic molecules in the blood products and then shine UV light so that actually it can kill or it can destroy the DNA so that actually the transmission is kind of like prevented. But the challenge with that, all those technologies actually available, that challenge is that actually, if you have pathogen reduction platelets, actually they need more of those needed for transfusion because of the platelet quality is kind of like decreased there. But another challenge is kind of as a transfusion as a whole concerned, like RBCs and whole blood, these pathogen reduction technologies are not useful therefore. So there is not a technology available yet for whole blood or RBCs for reduction of the pathogens or the bacteria. So can we improve it? Actually, that is kind of the questions actually like we looked at. So as I mentioned earlier, actually the current storage bags are kind of like polyvinyl chloride. These are hydrophobic surfaces. Actually, they are conducive of kind of like if you have a bacterial contamination, they can enhance or they can increase the biofilm formation, and that is kind of biofilms once formed. Actually, it is kind of like challenging to kind of to deal with that. And also kind of like because of this hydrophobic nature of the bag, the platelets or blood cells and other and plasma proteins can bind and activate and adhere to that actually. So that is also kind of like going to be a challenge. So we thought, if we can change the surface properties, because these bags are kind of like is available actually, like people have started using it actually, but there is not much improvement in this technology for the last 50 years or so actually. So we thought actually like if we could change the surface properties, is there any way actually like we could improve the storage quality of the platelets? And that is where actually like we thought actually, so can we generate or can we develop a kind of a super hydrophilic surface, and so that kind of like the protein adsorption is decreased or platelet adhesion is kind of decreased, so that is there any kind of like better chance or can we improve the storage quality? And then that is the story I'm going to talk about kind of the next couple of minutes on kind of the first part on development of anti-adhesive platelet storage bags actually. So improving the bag surface, can it improve the storage quality? So here actually like so our hypothesis is kind of reducing the platelet adhesion on the bag possibly can improve. So that is our hypothesis actually for that. So we wanted to kind of develop a technology or kind of like a techniques where it could be applicable for kind of like the storage bags. So using kind of like developing coating on kind of like a normal surfaces are pretty easy. But if you want to translate that to kind of a blood bag, actually like we need a lot of kind of things has to be considered there. So to do that, actually, like we started from kind of like starting from the bag coupons. So we cut the bag surface, the bag and the coupons, and then put it on kind of a 24-volt plates, and that is used for kind of screening the coating process or kind of coating components there. So we started from there actually. So then we kind of like we kind of like screen kind of like number of components, including kind of the polymers, including kind of what other kind of like components we can add. And then we add kind of like, we utilize the platelet concentrates to look at kind of like whether these coating components are kind of compatible with the surface or surface coating. And then look at kind of like, initial screening, actually looking at the activation state of the platelets and also kind of like the quantity of the platelets kind of adhered there. The goal is to kind of like identify the coating compositions which has got less platelet activation or platelet adhesion, and also kind of like less number of platelets actually adhered there. So we have this coating composition consists of kind of like several kind of like hydrophilic polymers, and in combination with the polydopamine actually, which is a binder used for kind of adhering these polymers on the surface of the bag. So in the initial screening, actually, we identified kind of like few different compositions, actually, which is for example, kind of like 3 different polymers in combinations with the binder, which shows kind of like really a good amount of like a significant decrease in platelet adhesion on the surface of the bag or surface of the coupons. So this give us kind of like compositions actually which you can kind of move to the bag surfaces. So then actually like we looked at kind of like, we applied, although we did kind of a lot of characterizations because of the time, actually I'm not going to show that data, but we transferred that kind of like whatever the codings actually we developed on the coupons and also the indexable plate. So we transferred it to kind of a 45 mil platelet bags, actually, which is a representation of kind of like a normal unit of the blood, platelet bag, which is kind of about 200 to 250 mils of the platelets. And then we adapted that to 45 mils and then stored the platelets for kind of 7 days to look at kind of like, whether the quality, of using this coding actually can be improved the quality of the, is there any improvement of the quality of the platelets? So what you are seeing here is kind of like, although we kind of like decrease the platelet adhesion or the platelet adhesion on the surface as we expected, but what we have seen that over the 7-day period, and we didn't see kind of a significant improvement in the platelet quality, even though the coding is kind of like doing its job as kind of like decreasing the platelet adhesion. So this is a kind of a little bit surprising to us, actually, like why. So you have, the bag surface is kind of like increasing the platelet activation, but it is when we do kind of the storage over 7 days period, and you don't see kind of any improvement there. But this coding actually shown to be kind of really, it is not activating the platelets, it is not adhering the platelets, it's a good coding, and it could be used for the blood bag materials. So that I'll come to that kind of like what are the other use of this kind of coding. So what you've seen that kind of like, although the platelet quality is not improved that much compared to the PVC bag, but the platelet adhesion is kind of like significantly decreased over the 7-day storage period, actually, which is suggesting that actually this coding is doing its job as it intended to do. So why then the question comes actually like why this kind of like although its adhesion is kind of, or activation is decreased, but why the quality of the storage is not improved. So one of the hypotheses that for the long time hypothesis in the field is kind of like the platelets kind of like activating on the surface, hydrophobic surface, due to the kind of like adhesion of the proteins such as fibrinogen and other proteins, and which is kind of like activating the platelets, and that is possibly the reason why the platelets are adhering more there. So we know that actually like we improved it, but why this kind of like this is not happening. So we utilized a proteomics approach to look at kind of like what kind of like proteins are adhered on the surface and see actually whether there's any changes between coded bag versus uncoded bag. Actually, so here, so we did kind of like a study actually. So we collected the platelet concentrates, and then we kind of like separated the plasma from there, and then we exposed it to kind of the coded bags as well as uncoded bag, and we looked at kind of like what kind of proteins are adhered onto the surface of the bag using kind of mass spec studies. So this is kind of the proteins actually what is on the bag surface, and looking at kind of 4 hours to a 7-day period. So what we found that kind of like total amount of proteins actually like is almost similar. But the most important thing is kind of like if you look at kind of the type of proteins, actually what type of proteins are adhered on the surface is shown that kind of like PVC versus kind of like one of the coded bag, you could see that kind of like there is a significant or kind of like there is a dramatic difference between the composition of the proteins adhered there. That is kind of like giving us kind of like our original hypothesis that actually like the protein changes could possibly improve it, but what we found that kind of like in the field actually like telling that actually like the bag surface is could be contributing to the deteriorating the platelet quality, but we found that actually like that is not the case, which is kind of like a eye-opening for us. Actually, like the coated surfaces or uncoated surface, it is not giving us kind of like good properties. But what we on this work actually shown that kind of like we can develop a better coated material or better coating, and can we utilize it for some other applications or some other challenges what is in transfusion area. So as I mentioned earlier, kind of like the bacterial contamination or bacterial growth is another important problems actually have to be dealt with that. So utilizing this coating, actually, can we develop a self-sterilizing bag? Actually, none of this bag material, none of this current blood bags have antibacterial properties. Actually, can we develop something utilizing, can we kill the bacteria at the collection point or kind of during the storage, so that actually like you don't have to worry about kind of the bacterial contamination or bacterial growth anymore? Actually, can we develop that? So that is where actually like we looked at kind of like he utilized a similar kind of like a coating process, and then we screened kind of the coatings again, and then we conjugated with antimicrobial peptide or antibiofilm peptides on the surface, and then we screened for kind of like better antimicrobial peptides or better antimicrobial coatings, and then and here actually like we are using a high throughput assays, and then looking at kind of like which type of coatings actually do have kind of antibacterial properties, as well as kind of like it's blood compatible there. So then we kind of like, our hope is to kind of identify kind of the compositions of the coatings which are kind of blood compatible, and then also broad spectrum active coating there. So here, in terms of like coating compositions, actually we already have identified coating compositions actually which are useful. So then we utilize kind of like antimicrobial or antibiofilm peptides actually, like which is kind of like interesting molecules actually which has got broad spectrum activity, and also kind of like you have seen that actually when these antimicrobial peptides actually attached on the surface, still they are able to kill the bacteria, unlike kind of like antibiotics, which are kind of like which could kill the bacteria, but when they are attached or on the surface, actually it is not going to be effective there. And also kind of like this type of peptides actually they have less prone to kind of resistance. So this has got kind of like broad spectrum activity there, less resistance, and also surface active, so that we could make the bags which are kind of non-leaching to antibacterial agents, and also kind of like it could possibly kind of kill the bacteria present there. When you are collecting the blood, actually, like you have kind of, even if it's a contamination, there is very, very little amount of bacteria there, so that actually there's a possibility, you don't need kind of that much antimicrobial activity for killing the bacteria there. So our goal is to kind of like, then we looked at kind of like, as I mentioned earlier, kind of like we had to identify kind of like which antimicrobial peptide or what composition of the antimicrobial peptide is kind of like going to be useful there. So what we kind of like looked at kind of like, this is a high throughput screen for antimicrobial peptides, and attached onto kind of the surface of the bag or the coupons, and looking at its activity there. So what we identified from the screen, there is kind of like a number of peptides actually which are able to kind of kill the bacteria quantitatively, and some of them are kind of like not effective. But this screen is allow us to kind of like identify kind of like some compositions which are really useful for the blood storage applications. So then we looked at kind of like, after screening, actually identified the bacteria. So here, identify the peptides. So we selected kind of a couple of them. Actually, we haven't analyzed all of them. Actually, we selected two of the peptides actually which are kind of like the top performers, and then looking at kind of like whether this surface compositions or codic compositions can effectively kill bacteria in platelet concentrates and plasma, as well as in kind of like an MSP actually, which is a bacterial, a bacterial growth medium. So what we have seen this here actually, like, this peptide coding can effectively kind of kill the bacteria in platelet concentrates, and also it can kill the bacteria in plasma, and also it can kill the bacteria in MSP2 actually. So that is showing that kind of like, this irrespective of the compositions of the media, and it could kill the bacteria there, which is kind of like a positive thing to kind of like move forward. And then we looked at kind of like, what is kind of like, one other important aspect of this for transfusion purpose, if you were to use, we had to make sure that kind of like this coding is non-leaching. Actually, the bacterial agents could not be kind of, it should not be leaching into the solution or leaching into the blood products. So that is another important criteria. So it has to kill bacteria in the blood, but also kind of like it should not be leaching into the solution. So there actually, like we looked at Nicholas actually did this work. Actually, he shown that kind of like one of this coding actually shown that kind of like on a surface it can kill bacteria in a staph epidermis, which is a kind of a very relevant bacteria for platelet storage, and it could kill the bacteria in platelet on a coded stage in plasma, but the leachate actually doesn't have kind of any antibacterial activity, which is suggesting that actually it is not leaching on the surface. And similarly, actually, like on platelet concentrate, you could see that actually it could kill the bacteria in the platelet concentrate, and also it could kill in whole blood too, actually. So that is suggesting that actually like it could be adaptable for not only for platelets, but also for kind of like whole blood as well as RBC storage. Then we did is kind of like, this coding actually adopted for kind of like a 5 ml mini bag. And then we kind of like did this kind of like studies in a long-term storage studies. Here actually like we have kind of like 2 peptide coated bags, and then you also have kind of like control bags, and we did have kind of like bacteria inoculated at 100 CFE per ml actually, which is at a higher concentration of bacteria actually than normally a kind of blood exposed in a transfusion conditions, and then we looked at these studies in up to 7 days actually, see how this performs. So what we have seen here actually, like this is for the kind of like storage for the bacterial growth, and you could see that actually like over 24 hours, it could kill, our coated bags can kill the bacteria, and our control bags actually like still grow bacteria kind of like normally as expected, and coated bags actually like, in platelet concentrates, it can kill that. And what about kind of the platelet activation or kind of like platelet quality? Actually, here you could see that control bags, with bacteria, the platelet quality is deteriorated, but the platelet activation levels are actually almost similar to the control bags, actually suggesting that this antimicrobial coating is effective in killing the bacteria without compromising the platelet quality there. And the platelet quality is also kind of like it's doing its job perfectly there. So in conclusion, actually, what we have shown that kind of like starting from a basic science point of view, actually, like can we develop kind of like better coatings, and what we showed that kind of like we could develop a platelet-friendly anti-adhesion coatings for a blood storage bag, and this can be started from a kind of a 90-series plate and we can adapt it to all the way to kind of a transfusion bag actually. So the technology is kind of like really adaptable there. So we demonstrated kind of a proof of concept self-sterilizing bags, and this using antimicrobial peptides and coatings actually could be potentially applicable for not only for platelet concentrate, but also for RBCs as well as whole blood. So we shown that kind of like high throughput screening is kind of like useful in identifying the better compositions. And also kind of like currently actually we shown kind of 45 ml bag as well as 5 ml bag. So we are currently kind of like scaling up this to a full plated bags as well as a whole blood storage bag, and also identifying what is happening, or kind of like more fundamental understanding is happening along with that. So before concluding, I would like to thank my extended group and also their talented postdocs and graduate students actually contributing this work. They are the major winners here actually here. And also my collaborators actually, extensive collaboration list, especially for this, the board letters actually shown they contributed to this this particular project, and also kind of like funding agencies actually who are supporting this work. And thank you very much for your attention. I'll be happy to answer your questions.
Hi, Jay. Thank you very much. Very interesting presentation. I have two questions. The first one, does the modification of the containers for platelet storage, does it affect the gas permeability?
So this coating is around 20 nanometers. Okay, so if you think about kind of very, very thin coatings actually, so we haven't seen any changes in the oxygen permeability. Oxygen measurements actually we have done is similar to the controls. There was no kind of like changes in the oxygen permeability there.
Okay. And also in one of your experiments, you show that effectively Staphylococcus epidermidis is killed in platelets, plasma, and media. But what I noticed is that in platelets, you needed a very small concentration of the peptide. So I'm wondering why in plasma, I think it was the lowest concentration that you tested. It was effective in killing in platelets, but not in plasma. You know why?
Yeah, I think, something that actually that concentrations actually what we are showing is kind of like solution concentrations actually used for surface conjugation. Okay, it is not effective concentrations on the surface actually I haven't shown. Okay, so that is kind of the modifications actually conditions actually used. On a surface actually it might be a similar concentrations actually. So what we have seen, but and also we had considered that actually like when the blood cells are present, there might be some changes actually on the activity of these peptides, and whole blood is actually we have slightly different compositions in compared to kind of like platelet concentrations or compared to the plasma there. Okay, so there is some changes we can expect.
Thank you, very interesting.
So can I ask this question? So you focus on bacteria. One of the big gaps we have with UV and amotocillin pathogen reduction is the inability to reduce titers of non-envelope viruses. Is your technology able to be paired with a defensin or an antiviral peptide that could be used to knock down levels of, like say, a hepatitis A or a hepatitis E virus in a platelet bag?
Yeah, that's a very, very interesting question, actually. So what I've shown here is kind of like a peptides, actually, which is antibacterial. But those technology for conjugation of the peptides, any peptides can be conjugated there, actually. So if there is a peptide which is, particularly good against viruses or kind of any other pathogens, actually, we can be. So we are also screening. We have the plans to screen kind of those peptides for antiviral activities too. So it is kind of like a broad spectrum antimicrobial agents, or it could be antibacterial or antiviral or other kind of pathogens actually, like it could possibly. And anti-biofilm peptides or antimicrobial peptides are shown to be active for against viruses as well as other things. So we could possibly use that.
Jay, Jay, enjoyed your presentation. Very interesting work. If I missed it, I'm sorry, but what temperature did you do this at? Cold stored platelets are a hot topic, if you'll excuse the expression right now. And there's a real push for actually using them and storing them at a much colder temperature. Have you, what temperature did you do this work at? And two, have you looked at potentially the differential effects certainly at the cold storages being used?
Yeah, so we did this platelet work at room temperature. Actually, that is the normal temperature. Actually, like used for the platelet storage, and we haven't tested the activity of these coatings at cold temperature yet. So we have because these peptides are kind of like surface active, okay, or it is kind of like it is interacting with the bacterial membranes differentially compared to the platelets. So it might be active at room, at cold temperature too, and also kind of like we plan to have kind of like this technology adapted to whole blood stored in kind of like at cold temperatures too. So possibly actually like it could work effectively there. So but we have to kind of look at that.
Super nice, Jay. A lot of these antibacterial peptides are pore-forming peptides. So presumably the ones that you're using are not pore-forming because they wouldn't be able to form these massive clusters and a pore while surface-bound. You wouldn't be able to get them in close proximity. So what's the mechanism of the antibacterial activity?
Yeah, so that's a very interesting question, Chris. So these peptides do have kind of multiple levels of action, actually. So normally, actually, like antimicrobial peptides, actually pore forming, actually, that is needed, kind of like these cells, has been interacting with the peptides. But these surface-active peptides actually has a different mechanism of action. For example, actually, like since it is kind of attached or conjugated on the surface, there is no leaching or there is no solution peptides. There is no pore-forming possibility there. So it is, what it does is kind of like it changes the membrane, if it kind of like interact with the bacteria or touches the bacteria, it changes the ion homeostasis actually. So that is sufficient enough to kind of kill the bacteria there. So it is kind of touch and kill mechanism actually, like that is sufficient. So that is kind of a cool thing with this kind of like technology where you don't have to worry about kind of like antibacterial agents in the blood product or any other product, but one touch is sufficient enough to kind of like kill the bacteria. So these peptides are kind of like in the range of like, this one of the peptides actually what we heard is a 12 amino acids to 15 amino acids. And some of them are kind of like, some of them are potent ones, or even 10 amino acids are good one too.
One more question, Karina.
Hi. Thank you so much. I'm just asking, can these antibacterial, the ones that are on the bag, can they activate like any monocytes, anything that's maybe remnants in the bag, or
Yeah, so this coding actually kind of really benign into that. Okay, so but we also had to consider that actually like you are killing the bacteria. Actually, the dead bacteria can do something there. But Sandra might agree to that. Actually, like when you have in a plated bag, actually when you're starting, you have maybe 6 CFI or kind of 5 or 6 colony forming units per bag is available. Okay, at that time. Okay, it's very very low concentration of the bacteria there. So if we can kill the bacteria at that time itself, there is no point of like there's a good chance of like it is not going to increase to 10 to the power 5 or 10 to the power 7. So there actually like is going to be a challenge for kind of killing that. So maybe like it may not be a big problem for bacterial debris because of very low concentration there.